that's because we start with a DNA molecule that is comprised of two chains, two independent chains, and when replication occurs, each of what is to be the parental strand is duplicated so that you have a sort of heterocomplex of old and new following DNA replication. And this old strand here, on the next round of DNA replication, you end up with something that looks like this in pattern. So it's semi-conservative. One strand remains associated in the new DNA, and one strand is newly synthesized. DNA replication happens at something known as a replication fork. So you have two strands, and those strands open for the initiation of DNA replication, and then the polymerase moves, opening wider the DNA replication fork through the activity of an enzyme known as helicase. And that elongation process occurs until the DNA molecule is copied. I'm not sure how well this shows up from the back of the room, but this is something I drew for one of my upcoming slides. And DNA replication always occurs at a loci, or a DNA sequence region, known as an ORI, which is spelled little o, little r, little I, all in italics. So when we're talking about chromosomal replication, that ORI is always called ORI-C. Just in case my accent makes me a little hard to understand. ORI-C. So that is origin for chromosomal replication. So we have worked out information about an origin of replication. Really, this was done, I guess, in the 1970s. So, origin of replication is an area of the chromosome. So we have a large molecule. In E. coli, it's circular. We have our little origin of replication, and we know that when the DNA melts in this area and DNA polymerase associates itself in that melted area, it moves in E. coli in two directions. So we call this bidirectional replication, and it results in two individual replication forks being produced in a single molecule. And this is actually possible to see doing transmission electron microscopy of purified E. coli genomic DNA. So here you can actually see the replication forks present here and here. And as replication proceeds, the structure is breaking down here a little bit, but we have newly synthesized DNA that is coming off from two areas on the molecule. How do we know that it moves in two different directions? Well, by doing the same type of experiment that was shown here with the TEM, but this time, rather than just detecting the DNA directly, we detect radiation. If we label E. coli that is being grown for 2 minutes with a low activity thymidine, and then follow that for 2 minutes with a high activity tritiated thymidine, and we examine that whole chromosomal molecule, if you spread that molecule out, you can see, if there was the origin of replication, we have weak signal incorporated and then strong signal incorporated into the new DNA, and that is demonstrable proof that DNA replication is going this direction and this direction because of the pattern of incorporation of the low activity tritiated thymidine and the high activity tritiated thymidine. Okay? Does that make sense to everybody? I see some faces in the back that look like they're puzzling. You want me to try and explain it again, or shall I move on? Nobody says anything that I've heard. Okay? So there was another way in which bidirectional replication was proved, and this was using a piece of viral DNA. So the virus SV40, too early for me to get my writing down, has an origin of replication on the DNA molecule. That's recognized. And on sort of the other side of that molecule, there exists an echo R1 site. So because DNA replication always begins at the exact same location, the origin of replication, we know that DNA replication is going to ensue from here. If we cut this DNA to linearize it, if the interaction of the polymerases creates the two replication forks, we'll be able to tell by the sort of anchor we have here with the cut. So if you look here, we've got a cut in this panel here that leaves us with a small replication, two small replication forks from the origin, right? And here is the same molecule cut in a slightly different way. And you can see from the length of this DNA and the length of that DNA relative to the cut that the origin of replication has been extended bidirectionally. Okay? So if this didn't convince you here, this is even easier to understand. Okay? So this was all proven in the 1970s. So replication, we know, is semi-discontinuous, which is an odd way of saying that DNA replication at the fork occurs differently depending on the strand that is being replicated. So when DNA replicase is running in this direction, it makes one leading strand and one lagging strand. It reads the leading strand and the lagging strand. And this lagging strand, because the polymerase, because the DNA is two strands, and the polymerase, because the way I've drawn this molecule, it's maybe not quite as clear as it should be. So let's make sure here in your minds the DNA is truly double-stranded. And so when this opens up like this, the polymerase is going to move along, and it's got to replicate both strands in order to keep the DNA double-stranded. So we have the leading strand and the lagging strand. The lagging strand gets made up of short little pieces of DNA, which have been termed Okazaki fragments due to the Japanese scientists that first identified them. And for each side of the bidirectional replication process, there is a leading strand and a lagging strand. Regardless of what you're going to talk about, in what kind of system, whether it's viral or cellular, DNA polymerases need something in which to add nucleotides on. So when we, as biologists, try to describe DNA to a chemist, The best way for us to do this is that we have amino sugars that are linked to one another by phosphodiester bonds. In short, to make a phosphodiester bond, we need an end with a phosphate and an end with a hydroxyl group to come together in a condensation reaction to produce that phosphodiester bond. Okay? So for DNA polymerase to make DNA, extend DNA, it has to have something with a free 3' hydroxyl group in order to add the 5' phosphate from the next nucleotide to create a phosphodiester bond. So cells, understanding what DNA polymerase need, have come up with a way of creating a starting point for DNA polymerase. RNA polymerase is very good at bringing in a ribonucleotide and start stringing them together. It doesn't need to be primed because of the way transcription works. So DNA replication relies, surprisingly, on short RNA transcripts. So those short RNA transcripts are made along the lagging strand, and those little transcripts each have a free 3' hydroxyl group, which enables DNA polymerase to start making the new copy of DNA. That creates, when DNA is being made, a long continuous strand, which is linked to its copy, and it creates short little fragments like this on the lagging strand. But this wouldn't work very well biologically. So DNA replication also has to involve the process of removing these tiny little RNA transcripts that are spaced between the synthesized pieces of DNA. So there is a DNA polymerase that will do this, and the last step in this process, so there's a DNA polymerase that will do this to remove these ends, converting them all to DNA. And then there's a third enzyme that comes in and acts on these gaps, known as DNA ligase. And it joins the little Okazaki fragments on the lagging strand together so that we have a complete semi-conservative DNA replication process. So here we have the initiation of DNA synthesis. The DNA is unwound by an enzyme called helicase. We have a small RNA primer that is laid down, and that enables DNA polymerase to begin to copy. The helicase that is associated with this large enzyme complex then unwinds the DNA, extending this replication fork, and the leading strand will continue to be synthesized. Now, in this particular system, it may become more obvious what some of the problems are with the lagging strand synthesis. So we've got 5' and 3' and 5' and 3' on the parental copies. And so the new DNA being synthesized here, this is its 5' end, and DNA has to be synthesized, RNA too, in a 5' to 3' basis. That creates an issue. Yes, Sam?
Why would you use RNA
because DNA polymerase cannot, no DNA polymerase works by initiating DNA synthesis without having a free 3' hydroxyl group to attach a phosphate, the phosphate from a free deoxy nucleotide. It just can't do it. There isn't one that we know of that can do that. So there has to be a substrate for that phosphate to bind. And the only way that biology has come up with a way of doing that is to use a short RNA primer, which is produced through transcription. So there's something RNA polymerase can do in that respect that no DNA polymerase can. Okay? So the problem here is that the lagging strand has to be polymerized in this movement, but the polymerase itself is moving this way. So nature solves that by these lagging strands. So we have an RNA primer laid down and DNA polymerase extends it. And this sort of works just the way I described it on the board. So as this initiation complex, polymerase complex moves down, opening the replication fork, these lagging strands are fused together by first removing the RNA primer, by breaking it down with an exonuclease function, and then an enzyme comes in that ligates new DNA together. Of course, it was proved about these lagging and leading strands. And to do that, a bacteriophage, this is a bacterial virus, it's got a double-stranded DNA genome, and this T4 virus, the DNA that was being produced within an infected E. coli was pulse-labeled with tritiated thymidine for a few seconds. And then the DNA that was obtained was, through centrifugation, pulled through a gradient that separated small fragments and large fragments. And if you then take these fragments, not only can you separate them on a gradient, but you can also separate them via a sort of pH gradient. And so these Okazaki fragments were, in the end, identified by looking at 2 isogenic mutants. Okay, so I'm using a word I'm not expecting you to know. Does anybody know isogenic? Isogenic mutants are incredibly important for being able to do biology research. An isogenic mutant takes a parental something or other, in this case T4, and a distinct relative, clonal relative, of the parent that has one gene changed. So it is absolutely identical to the parent, except one gene is changed. So it's isogenic. It is identical genetically except for one thing. And those are crucial. These types of mutants are crucial for being able to work out biological functions of genes, because you can't compare 2 unrelated strains. They may be the same type of organism, but they could have multiple differences in their genetic makeup. But by using an isogenic mutant, you are ensuring that the genetic background is identical except for the gene of interest. And in this particular case, T4 has provided molecular biologists for decades with the best DNA ligase, highest specific activity known. And the gene 30 of T4, bacteriophage, is a DNA ligase. So by looking at phage DNA that was produced during a wild-type T4 infection or a ligase isogenic mutant infection, we can see that this peak here is very short fragments of DNA, and these are long pieces of DNA. And in the mutant, in the absence of a ligase, we see that T4, most of the DNA that gets made are these short fragments. So this was proof that these short fragments that get made on the lagging strand are ligated and joined together in the process of DNA replication. So as you might imagine, the DNA polymerase complex that drives leading strand replication and lagging strand replication at the replication fork is rather complicated. Okay? There's a lot of functions to be performed. So, the first thing that has to happen is that the DNA helicase has to open up and unwind the DNA to produce a fork in the first place. Okay? The next thing that has to happen is that a DNA primase, which is another word for an RNA polymerase that makes a short transcript. So this enzyme is never associated with the production of mRNA or rRNA or tRNA. It just makes short little RNA transcripts for DNA polymerase in what is not a sequence-dependent fashion. So DNA polymerase recognizes DNA and makes a very short transcript, and when that transcript is made, the DNA polymerase holoenzyme, DNA polymerase 3, will begin the leading strand synthesis. Now, this creates some problems. So as helicase is unwinding the double-stranded DNA at the replication fork here, it's creating two short fragments, two short pieces of single-stranded DNA. Nobody likes single-stranded DNA. Single-stranded DNA is a sign of viral infection. It can activate systems in the cell to destroy single-stranded DNA. So part of the process of replication involves single-stranded binding proteins, which will immediately bind to the opened DNA, protecting it from degradation. And these single-stranded binding proteins then are removed in order for DNA primase to come in on the lagging strand, make a short transcript from which DNA polymerase 3 will perform a replication process in this direction. So if the enzyme is located here, how in the hell can it work in that direction too? And the secret to this is that replication does not happen in this type of plane. So when this enzyme is moving, and this will make sense a little later on when I show you a movie of everything functioning, the DNA that is synthesized on the lagging strand gets tied in almost like a bow. So it gets looped back on itself, so that the DNA that is being synthesized here is actually pointing in this direction when it's made. But you only make short fragments. And in order to be able to put all of this together, we need So this is DNA polymerase III. It's the major workhouse for DNA chromosomal replication, but it also requires another DNA polymerase known as DNA polymerase I. DNA polymerase I is a very small polymerase with kind of three functions. It can synthesize short fragments of DNA through a polymerase function, but it can also chew back the RNA primer, removing it. It can proofread, so it helps find if there are nucleotide mismatches, and in chewing off the primer, it leaves, and replacing it, it puts a 5' phosphate end right next to a free 3' hydroxyl group from the product of DNA polymerase III, and then these two products can be joined together by DNA ligase. Okay? Don't give up on me yet. I'm going to see if I can make all of this come to life. So, I don't really think that this slide is got anything that I want to say right now. So we'll come back to that. Let's focus on the first part of DNA replication. And that is the need to get the DNA